Okay, good morning everyone. Welcome to lecture 26 of BMS 300. I see that you're all in high spirits, so I think that's a good sign. So today we'll be wrapping up with the nervous system, and I'll be talking about the autonomic nervous system, and I think it should take me around 15 minutes to get through this material, and then we'll go through the key questions at the end of each of the lectures. We'll go through that together, all right? And tomorrow we'll do the muscular system, and then in the remaining time, maybe for 40 minutes we'll do the muscular system, and in the remaining time we'll do some polar questions on the nervous system. Yes?
Why don't you just turn up the volume a little bit?
Okay. Is that better? Okay. Okay. Did you hear what I said earlier though? Alright. So with that, let's get started. Here's an overview of what we'll be discussing. So we're going to talk about the differences in responses between the sympathetic and the parasympathetic nervous system. So we'll find that the autonomic nervous system can have two types of divisions, the sympathetic and the parasympathetic divisions. So the sympathetic divisions is going to be regulating the fight and flight response, where there's heightened mental alertness and so on. Whereas the parasympathetic division is going to deal with the rest and digest, feed and breed response. And we will spend a few minutes talking about what are the neural pathways involved in carrying out the sympathetic versus the parasympathetic response. And we will tie into the concepts like what is ganglia, the synapse, and nuclei. So we will be building on those concepts. So notice here that the sympathetic nervous system is shown here in red. So you have a greater divergence in how these neurons are branching off, whereas in the parasympathetic division it's going to be less divergent. And it's sort of color coded. This guy's wearing a blue shirt and resting in the region allocated to the parasympathetic nervous system is shown here in blue, and the part allocated to the sympathetic system is shown in red. And then we'll move on to talk about the types of neurotransmitters which is released in the sympathetic versus the parasympathetic systems. And finally, we'll talk about how the G protein coupled receptors, which I mentioned previously, how they tie into the activation of the autonomic response. So we will talk about three types of pathways. So in G protein coupled receptors, you can have three complexes, and one complex can have the alpha S subunit, another type of complex can have the alpha Q subunit, and the other one can have the alpha I subunits. And notice that for each of these subunits to be released and to trigger the downstream mechanism, the hormones will be binding to various receptors. Now the names of those receptors, I don't want to mention it because I don't want to add to the existing complexities. Right? Know that there are different receptors which the hormones can bind to, and depending on the receptor, you could either have this G alpha S pathway, G alpha I, or the G alpha Q pathways. So what is the autonomic nervous system? Right? So this is the part of the nervous system which is responsible for involuntary control, and it's going to control various organ systems we will consider in the future, such as the cardiovascular system, the respiratory system, the urinary system, digestive system, and also the reproductive system. So what's happening in these systems is much of it is not under our control. And for example, in the digestive system, once you chew and swallow the food, after that you have no more control until it comes to defecate or urinate. Right? So there's only some parts of it we can control, but the majority of the functioning of these systems is under the control of the autonomic nervous system. And so the overall purpose is to adjust the internal water concentration, the electrolyte, nutrient, and dissolved gas concentrations in the body. Right? So that is the goal of this autonomic nervous system. So we find that the signals from the brain are going to be carried out to the ganglia shown here, and from the ganglia goes to the peripheral target organs. I mentioned previously what ganglia refers to. It's just the collection of neuronal cell bodies outside the central nervous system. So it's referring to the neuronal cell body collection in the peripheral nervous system. And from there, it goes to various target organs. So the two major subdivisions of the autonomic nervous system are the sympathetic division, which will regulate the fight or flight response, and the parasympathetic division, which can mediate the rest or digest response, also the feed and breathe, right? So both of those should happen under the activation of the parasympathetic nervous system under restful conditions. So now we will see what are some of the responses which can be observed when you have an increase in sympathetic activity versus the increase in parasympathetic activity. So I'd like to explain these responses by telling you a short story of what happened to me. Right. So a few years ago, I was driving back from Cleveland to Ithaca, right? And in Cleveland, I spent time with my sister's family. My parents were visiting from out of the country, so it was a nice family reunion. So obviously, probably ate a lot of food. And as I'm driving back, it's around 7:30 p.m. and it's getting closer to twilight, right? And I'm feeling drowsy. So this was before the time when I discovered the joys of listening to audiobooks and staying awake. So I was trying to drink coffee. I was chewing on ice. Nothing was really working. Then as I was going along, I noticed on the right side of the road there was a deer crossing, right? And as I got closer, I realized there was a deer on the left side as well. So one behind the other, and I went right in between the two. So no surprises that the moment I got past the deer, I instantly had heightened mental alertness. Right? What the coffee couldn't accomplish, what chewing on ice couldn't accomplish, those two deer accomplished like in an instant. And no surprises, you will have an increase in metabolic rate, meaning you're starting to burn more calories to fuel whatever you need to do to run away from a situation. You'll also have a decrease in digestive and urinary functions. So in terms of defecation and all that, you prefer to go to a more comfortable place rather than going out in the forest, right? So when you have this sympathetic activity of decrease in digestive and urinary functions, and that's the reason why they say don't just eat while you're stressed. You have to be nice and calm and relaxed. There will also be an increase in the respiratory rate when you're having this sympathetic activation, and the respiratory passageways will start dilating so that you can get more oxygen into the lungs. And your heart rate and blood pressure will shoot up. The reason for that is you want the blood to be circulated at a faster rate so that the tissues in your body can get the oxygen, and also carbon dioxide can be removed from the various tissues. And this, any energy reserves like fats, they'll be broken down when you have this sympathetic activation. So even when you're exercising, you're using these energy reserves and you're burning more calories. And finally, you will also have an activation of the sweat glands as well. So anybody recognize this picture here? Recognize it? So last year there was a bear on the oval, right? So if you were to encounter this bear, you also may have had increased sympathetic activity, right? So let's see what are the responses during increased parasympathetic activity. So this is what I would have been experiencing before I drove between those two deer. Right? You have a decrease in metabolic rate. You'll have a lower heart rate and blood pressure because you're all relaxed. Hopefully you're all having this increased parasympathetic activity right now. And then there will be increase in salivary secretions and also the secretion of various digestive glands. And this is important for helping optimal digestion, as we will see in the digestive system. And the motility of the digestive tract will increase as well. So the digestive tract is made up of, is lined by smooth muscles, and so they will undergo this rhythmic contraction and relaxation to move the food you ingest all through the digestive passageway. So that is what is meant by an increase in motility of the digestive tract. And there will be a greater blood flow to the digestive system to enhance the digestion. And also you will have a stimulation of urination and defecation. Right? So when you see this line here, you may wonder, why is it that sometimes when people are really scared, they may urinate or defecate without their self-control, right? That is something which is beyond the scope. When you take a more advanced course, you will learn about the various parts in the brain which are controlling this, and when you're really scared, you can sort of override those mechanisms which control urination and defecation. Okay, so that's a topic for another day and another advanced course. So any questions at all on what responses you see in heightened activation of parasympathetic and sympathetic divisions? Okay, if there are no more questions, let's move on to this neural pathways. Right? So color coded here for your convenience is the sympathetic nervous system in light green. So this gray part is showing you the central nervous system, and we find there is this preganglionic neuron which is moving into the peripheral nervous system. And this preganglionic neuron is going to release acetylcholine in its synapse. And this preganglionic neuron will synapse with the postganglionic neuron, which will in turn go all the way to the target tissue, or maybe be in close vicinity to the target tissue. And this postganglionic neuron can release epinephrine or norepinephrine. Right? Now, when you look at the parasympathetic nervous system generation, it's shown here in purple for your convenience. Notice that the preganglionic neuron is also releasing acetylcholine, right, in the target tissue, and there's also a postganglionic neuron which also releases acetylcholine. But keep in mind that the acetylcholine which is released by this preganglionic neuron will bind to ligand-gated ion channels. Right? These ligand-gated ion channels also are known as these nicotinic receptors. Whereas you have the muscarinic receptors where you have G protein coupled receptors being activated. So even though acetylcholine is released both by the preganglionic and the postganglionic neuron in the parasympathetic nervous system, in one case it is activating ligand-gated ion channels, in the other case it will activate the G protein coupled receptors. And that's why we will be talking about these various types of G protein coupled receptor pathways later in the lecture. So now let's look at some more about this type of innervation. Right? So on the left side, we will be talking about some of the characteristics of these preganglionic and the postganglionic neurons in the sympathetic division. So the preganglionic neurons, notice that their fibers are a lot shorter, whereas in the peripheral nervous system they tend to be a lot, in the parasympathetic nervous system they tend to be a lot longer. And these will release acetylcholine, right? And the acetylcholine will bind to ligand-gated ion channels. Whereas the postganglionic neurons, you will see that the cell body is going to be quite far from the target tissue. And as a result, the nerve fibers from these postganglionic neurons will be very long, or it's going to release the epinephrine and the norepinephrine into the bloodstream, and that's how it gets to the target tissue. So please keep in mind that epinephrine and norepinephrine are both hormones, so they will be transported through the bloodstream to get to the target organs as well. Now on the other side, we find that the preganglionic neurons in the parasympathetic division, they tend to be a lot longer than the ones found in the sympathetic nervous system. And they will also release the acetylcholine in the synapse, and that binds to ligand-gated ion channels. And the postganglionic neuron, shown here in gray, it's going to be very close to the target tissue, or it will be found in the target tissue. And so the nerve fibers here are much shorter, and it's going to release acetylcholine, which will activate G protein coupled receptors. Any questions at all on this neuronal pathways for sympathetic and parasympathetic divisions? Okay, if there are no questions, let me just go over these points in the context of these two figures. Okay, so we find that these preganglionic neurons in the sympathetic division, the cell body will be found in the spinal cord. Notice here it is shown here in red that the spinal cord is where the cell bodies for these preganglionic neurons in the sympathetic division are found. Whereas in the parasympathetic division, we find that some of these cell bodies may be found in the brainstem, and the others are found at the caudal or the tail end of the spinal cord. So if you think about where these cell bodies are, it's going to be alongside the sympathetic nervous system division. Right? So it's going to be found on either side of the sympathetic nervous system, where you have the neuronal cell bodies for the parasympathetic division. And notice that this red line is denoting the preganglionic fibers, whereas the black line is denoting the postganglionic fibers. And in the sympathetic division, you notice that these red lines, they're going to be much shorter than the ones on the right side in the parasympathetic division. So the nerve fibers will be much shorter in the sympathetic division, and in the parasympathetic they are much longer. And the postganglionic neurons, their cell bodies will be a lot farther from the target tissue. That's why all of these black lines indicating the postganglionic fibers tend to be longer, whereas in the parasympathetic division they are pretty short. They're right nearby the target organs, as you can see right here. And yeah, so this is just pointing out that the nerve fibers are longer for the postganglionic neurons in the sympathetic division, whereas the postganglionic fibers in the parasympathetic division are much shorter. Now you'll also notice that there is going to be greater divergence in the sympathetic division, and what we mean by what is the functional significance of this greater divergence is that once the sympathetic system has been activated, multiple organs are going to be triggered at the same time. In an instant you have increased heart rate, increased breathing rate, so multiple organ systems are activated at the same time for the sympathetic division. Whereas for the parasympathetic division, you can have just a few organs activated, where maybe you're focusing your eyes on something and your hands is working on something very minute and delicate. So you could have fewer organs being activated during the parasympathetic activation. Also, just to remind you that in the target tissue, you have acetylcholine, which is acting when the parasympathetic division is activated, but you have epinephrine or non-epinephrine acting on the target tissue in response to sympathetic activation. And keep in mind that most vital organs, they will have a dual innervation. They will have innervation both from the sympathetic and the parasympathetic division, right? And that will help to establish what is known as an autonomic tone. So this autonomic tone is maintained by these neurons, which will have a background level activity even without any stimulation, right? And the importance of this is to have a regulatory effect. So imagine that there's a car, right, and the car is turned off. All you can do is turn it on. So it can go from either off to on. But if you're driving the car at a certain speed, you can go faster or slower, right? So similarly, you have this background level of both sympathetic and parasympathetic activation on certain organs, so that when needed, you can either increase one activation versus the other. So an example of this is the cardiac muscle. You have both epinephrine and norepinephrine being continuously released even when at rest. And when you're in resting conditions, the parasympathetic division will dominate, but when you have stressful conditions, the sympathetic stimulation will take place and more epinephrine will be released. So this is how you have the autonomic tone maintained for major organs. So when we get into the other organ systems, we will talk about how the autonomic nervous system can control their function. So now let's move on to the G protein coupled receptors and what pathways they activate. So let me just go through this figure first, which you're already familiar with. So I told you that the hormone is going to be the first messenger, and the reason the hormone is binding to a protein receptor is because that hormone is hydrophilic and it cannot get past the lipid bilayer. So once it binds to this protein receptor, it's going to activate this G protein coupled receptor complex, right? And in this figure here, you just see one blue box saying G protein being activated, but in reality, you have three different subunits. You have an alpha, beta, and a gamma subunit. And so this alpha subunit, when it's activated, it will cause this adenyl cyclase to be activated, which will turn ATP into cyclic AMP, and that can have further downstream effects. Right? So this is the overview of what we discussed previously. But keep in mind that depending on what receptor the hormone binds to, there can be many types of receptors, and depending on what receptor the hormone binds to, it can either cause the release of a G-alpha S subunit, or in the case of another receptor, it could cause the release of G-alpha I, or in another case, it could cause the release of G-alpha Q. Right? So what is the significance of G-alpha S versus G-alpha I versus G-alpha Q? So both G-alpha S and G-alpha I both are capable of binding to adenyl cyclase. So the enzyme which converts ATP into cyclic AMP can be bound by G-alpha S and G-alpha I. So when G-alpha S binds, G-alpha S, the S subscript stands for stimulatory. Right? So G-alpha S has a stimulatory effect, and it will cause the adenyl cyclase to be activated, and as a result, you have more cyclic AMP being produced. And when you have more cyclic AMP can activate protein kinase A. And in this figure, they have just mentioned it in general as kinase. Right? And once protein kinase A is activated, it can add a phosphate group onto various molecules, and that can either increase or decrease cellular activity as needed. And here you find that some of the cellular activity could include opening of ion channels or activating enzymes. And opening of ion channels is of relevance to us in the nervous system because any sort of signals for the brain for the central nervous system to detect, there has to be the movement of ions across channels. Now, G-alpha S is going to have an inhibitory effect, and it does the opposite of what G-alpha S does. So this will bind to adenyl cyclase, and it will decrease the cyclic AMP production, and as a result, it can decrease the protein kinase A synthesis as well. So is this clear how G-alpha S and G-alpha I have different functions? And keep in mind it's all mediated by epinephrine and norepinephrine. Where it changes is the protein receptor could be different, and depending on the protein receptor, you could have either G-alpha S or G-alpha I. Now let's move on to what G-alpha Q does. Okay. And G-alpha Q is going to activate the IP3 pathway where you have the inositol 3-phosphate. And this is the figure which I haven't shown you previously, and I like this representation a lot better than the one I showed you previously for how intercellular calcium is going to be released. I mentioned that cyclic AMP is one type of secondary messenger. Calcium releasing inside the cell is also a second type of secondary messenger. So this alpha Q subunit will bind to phospholipase C, so that's a type of enzyme, and that will convert PIP2 into IP3. So PIP2, it's sufficient if you know that term. It stands for phosphatidylinositol bisphosphate, but it will convert it into inositol triphosphate, right, which is IP3. And IP3 will in turn bind to receptors in the endoplasmic reticulum, and thereby can cause the intracellular calcium to be released. And one of the products in addition to IP3 is also diacylglycerol PAG, which can in turn bind to protein kinase C. And the subscript C in protein kinase C is standing for that it needs calcium to be activated. So the intracellular calcium can bind to protein kinase C, and the activated protein kinase C can phosphorylate more substrates. So we see that as a result of G-alpha Q activating phospholipase C, it causes more calcium to be released in the cytoplasm, and this calcium can also cause the contraction of smooth muscles as well. How exactly smooth muscle contraction is regulated by calcium, we will discuss in the next set of lectures. Right? Any questions at all on how G-alpha S, G-alpha I, and G-alpha Q carry out their downstream effects? The bottom line is G-alpha S is going to increase cyclic AMP, G-alpha I will decrease cyclic AMP, G-alpha Q will cause calcium to be released from the intracellular stores. That's the level of detail I hold you responsible for. Questions, concerns? Okay, there are no other questions. Let me just summarize what we've been discussing in part 5. So we talked about responses of the sympathetic activity, and that's going to focus on short-term goals, like running away from a specific danger, and the parasympathetic activity is going to focus on long-term goals. Right? So if you're really stressed out, you're under sympathetic activation, whereas if you're relaxed, you have the parasympathetic division being activated. And we also saw how the sympathetic division has got greater divergence, and this means that more organs will be activated when you have the sympathetic division being activated, whereas we can have few organs being controlled by the parasympathetic activation. And acetylcholine is the neurotransmitter which is released by the preganglionic fibers, both in the sympathetic and the parasympathetic division, whereas in the parasympathetic division you have acetylcholine being released by the postganglionic fibers, which will bind to G protein coupled receptors. On the other hand, the postganglionic fibers in the sympathetic nervous system will release epinephrine or norepinephrine. Okay? And we also talked about the 3 pathways of G-alpha S, G-alpha I, and G-alpha Q. And I told you that G-alpha S will increase cyclic AMP, G-alpha I will decrease cyclic AMP, and G-alpha Q can cause an increase in intracellular calcium concentrations. Yes?
Just double checking, so the G-alpha S, I, and Q are all released as a result of the binding of acetylcholine in the post-ganglion.
Correct, yes.
Okay.
And acetylcholine post-ganglion, yes.
Okay, and that's only during the parasympathetic.
It's also happening during the sympathetic as well.
Okay.
Yeah, if you remember part 1 of the endocrine system, I told you that there are secondary messengers activated, and I told you that epinephrine and norepinephrine can cause an increase in cyclic AMP activity, and cause a decrease in cyclic AMP activity, and also cause an increase in calcium release as well.
Gotcha. Okay.
Yeah.
Thank you.
Yeah, thank you. That's a good question, right? So epinephrine can activate G protein coupled receptors as well. That's something we discussed in part 1 of the endocrine system. So what I want to do for this